Hey guys, so in this video, I have my friend here, Sam Witt, who is an AP Bio student. I figured I've got a few practice problems that have to do with AP Bio, the experiment question that you'll get. And so we've got 5 questions. I'll show you where they're from. And yeah, so first of all, make sure you, if you want to try them, pause the video, either go to the AP website or just look at them on the screen here. I figured it would be easier also to explain and work with an actual student than to sort of just talk to the computer screen. So with that being said, I'm going to share my screen and we will look at these AP Bio practice questions. Again, these won't be exactly the same as the format that will be on your question 1, because the question 1 on the 2020 exam will include a lot more information. But the basic experiment portion of that question will be very similar. In fact, the two that the AP released have been very similar to these kind of questions. So let's start here, right? We've got a graph here, and we'll look at sort of our situation first. So the common bed bug is a species of insect that is being increasingly resistant to insecticides. Bed bugs possess several genes suspected of contributing to the resistance, including P450, ABCA, and CPS. To investigate the role of these genes in insecticide resistance, researchers deleted one or more of these genes in different strains of bed bugs, as indicated in Figure 1, and treated these strains with the insecticide beta-ciflurthrin. AP Bio exam generally has some difficult words to pronounce. Each strain was genetically identical except for the genes and was equally fit in the absence of the insecticide. The percent survival of each strain following the insecticide treatment is shown in Figure 1. So we've got different strains. And it says percent survival of the 5 strains of bed bugs treated with the insecticide. Plus indicates the gene is present, minus indicates the gene is deleted. Error bars represent the 95% confidence interval, right? And so what the error bars, which are really important, and there will be a lot of questions in here that have the error bars. If the error bars are not overlapping, you can know that there's a statistical difference, there's a significant statistical difference between the data collected, right? So if we look at numbers, strains 2 and 3, because the error bars overlap, there's actually no significant statistical difference between this data. They could be the same, right? The means of the data are different, but because of the uncertainty, they really could be the same actual result, right? So let's look at A, and remember, if you, hopefully you'll pause this video and try this on your own. So we, letter A tells us to identify the control strain in the experiment and use the means and confidence intervals in Figure 1 to justify the claim that ABC8 is effective at providing resistance to beta-ciflurthrin. So we know that the control strain, right, is the thing that we don't change. Right, and so if we look at these, which one appears to be the control strain, 1 through 5, would you say?
So it'll be the one without anything deleted.
Correct, right, because that's the one that we don't change. And we're going to be comparing things to the control. So now we want to use the means and confidence intervals from Figure 1 to justify the claim that ABC8 is effective at providing resistance to beta-ciflurthrin. So what two groups should we be comparing if we're trying to justify the claim that ABC8 is effective at providing resistance?
So it should be the control strain, and then the one where the ABC was deleted.
Correct, right, so 1 and 3. And the justification that we'll give, right, number 1 is, the first thing to note is that they are statistically different, right? They're significantly different because their error bars do not overlap, right? That alone could even earn you a point on this AP Bio exam. You have to acknowledge that. And then, right, it's the percent survival is far lower, right? Is significantly lower, right? We can use that because it's significant because of the error bars. So number 3, when ABC8 is deleted, it is significantly lower than the control where everything's there, right? So that's how we would justify that. Now, here's a nice question: why can't we compare strains 1 and strain 5?
There's too many things that are different from
Correct.
the control.
Right, you can't change more than one thing. And this one may be a little bit harder. Why can't we compare? I mean, evidently it would work, but what might be wrong with comparing strains 2 and strain 5?
Because strain 2 doesn't have all of them there.
Right.
You could, but
if we compare it, I mean, there's only one thing different. So the main problem with doing that, right, is potentially you could have some kind of interaction factor that maybe, and this is sometimes even possible, ABC8 alone, right, if we just looked at 2 and 5, ABC8 alone may not actually make a difference. It may only make a difference when combined with P450, right? So that's why we really need to use the control. B, P450 encodes an enzyme that detoxifies insecticides, right? And so this is, again, we're going to go back to enzyme activity in proteins, right? ABC8 encodes a transporter protein that pumps insecticides out of cells. CPS encodes an external structure protein located in the exoskeleton that greatly, which is sort of it's the skeleton sort of outside of the main part in bugs, right? That greatly reduces the absorption of insecticides. Based on this information and the data in Figure 1, explain how a deletion of both P450 and ABC8 results in lower survival in bed bugs compared with the deletion of CPS only, right? So, the first thing that we can notice is group 4, right, is definitely very different from group 5, right? And we're trying to explain why is this data different in group 5 different from this data in group 4, right? And so what we can notice is the things that really help, right? The things that make the survival rate a lot bigger, P450 and ABC8, CPS does not do that quite as well. So we have to think of a possible biological reason why, right? And so let's start by looking at strain 5, right? And I'm going to read the exact answer that the College Board gives, and then we can sort of look at that and see how we could get to that. So it says here, bed bugs can neither detoxify or pump out insecticide, right? Which results in a lower chance of bed bug survival, right? So even if there's a little bit, right, in group 5, even though there's, a little bit less that might get into the bed bug because the absorption is lower, it can't pump it out, right? So it, needs, it will lower the survival. But how about group 4? Even when it doesn't have this protein that lowers the absorption rate, it still has a pretty high chance of survival, actually not even statistically different from the control. And the answer, one of the things you could note there is that if you don't have CPS, even though the insecticide can get into the bed bug, it still gets pumped out, right? So you can show that maybe even potentially P450 and ABC8 are incredibly effective at pumping out the insecticide, while right the combination of those two, both pumping out and detoxifying. But strain 5, right, CPS, when you don't have it, even though it lets it in, it you know, you can still pump it out and detoxify it, but when you, even when you don't let it in, CPS, I guess, is not perfectly effective at keeping the insecticide out, obviously, right? So it's using sort of biological reasoning, understanding of what these kind of functions with these proteins do. Okay, so hopefully that problem makes sense, and we'll go on to the next one. So here we have another thing with confidence intervals. And the first thing you might notice on this is none of the data, right? That's the first thing you might look here. None of the data seems actually statistically different. There's no significant difference from any of this data, right? So let's look at our scenario first, and you can pause this video and try it. Researchers conducted a study to investigate the effect of exercise on the release of prolactin in the blood. The researchers measured the concentration of prolactin in the blood of 8 adult males before and after 1 hour of vigorous exercise. As a control, the researchers measured the concentration of blood prolactin in the same group of individuals at the same times of day 1 week later, but without having them exercise. The results are shown in figure 1, right? And it says the data represents the means plus or minus 2 standard error of the mean. So that's the measure of uncertainty. They're not using the standard deviation. They're using the standard error of the mean, right? And they're doing plus or minus 2 standard error of the mean to show the uncertainty in this data, right? And so it's sort of just understanding qualitatively what the standard error of the mean is sort of talking about. So first is justify the use of the without exercise treatment is the control, right? And so, right, when we're talking about a control, the main idea of a control is what?
So you can compare it with the ones that aren't.
Right. And so it's something that you're going to basically not be changing. You're not going to do anything intentionally to sort of, you know, try to get a response, right? And so we're looking at does exercise affect blood prolactin level, right? That's what we're asking. We're asking, does exercise affect this? So we can't just look at what happens when we exercise. We have to look at, well, what would happen if we didn't exercise? What is the normal situation, right? The normal situation would be without exercise. Then you're doing something in the experiment. You're exercising, you're doing something, and you get results, right? So then you can compare. So our justification, so that would be one way to justify. Another idea is that and here's exactly what the College Board says, that we use the control to make sure that the results that we get, right, we're only changing one thing, right? And because we're only changing one thing, we can be sure that if we got a different results, that it is only based on exercise. There's nothing else going on, right, that changes between these two trials. Right? So it goes back to what you were saying, right, with needing to control something, right? And so that is sort of a way that we would justify really the need for control in general, right? So B, using evidence from the specific treatments, determine whether prolactin release changes after exercise, justify your answer. So what would you think if, how would you think that we would answer this question?
Well, what I'm looking at is that with exercise, 0 hours is the same as exercising for 1 hour.
Right.
So, and they both went up. So even if you exercise 0 hours is nothing, it's still on up for some reason.
Right. And but the thing we have to remember though, right? And this is sort of the trick of this question is all of this data is statistically the same. Right? We actually, because of the error bars, we cannot tell the difference between the data, right? So the answer that they want is so we're looking at sort of after exercise. All of this data is statistically the same. The error bars, the plus or minus 2 SEX bars are all overlapping. So there's actually no change.
Doesn't do anything.
Right. There's no change. So we cannot conclude, right? Or we can determine that prolactin release does not changes. It does not change. Prolactin release does not change after exercise because the data that we're getting is statistically the same. Right? And you can compare, for example, comparing the 1 hour bar with this 1 hour bar, right? So we're only changing one thing, whether or not it exercises, or you could change the this T equals 0 bar with this T equals 0 bar, right? You just have to compare two of those bars and say that there's no statistical difference because the error bars are overlapping. Okay. So now we have one. We don't have a graph here, but we will have to talk about controls and different experimental groups. So this is 2015 question 5. Phototrophism in plants is a response in which a plant shoot grows toward a light source. The results of 5 different experimental treatments from classic investigations of phototrophism are shown above. And so the first thing to look at is, right, the first thing we can see is what is the control? And it should be pretty obvious here.
The unaltered.
The unaltered, right? We're not changing anything. So let's look at this. A, give support for the claim that the cells located in the tip of the plant shoot detect the light by comparing the results from treatment group 1 with the results from treatment group 2 and treatment group 3. So they're actually being very nice to us. They're telling us exactly what to compare, right? Even though we should know that we would have to compare groups 1 with something, right? Because it's the control. Now, if we look in 2 and 3, right, both of these show no phototrophism, right? They're both completely straight. They don't bend towards the light source, right? And the difference between so let's first look at groups 1 and 2. The difference is group 1 has a tip, group 2 does not have a tip, right? In group 1, and they're different, right? So we can first conclude that the tip is the part that controls this, right? Because the only thing different between groups 1 and 2 is whether or not they have a tip, right? So the tip, we can conclude that the tip is what's responsible for the phototrophism response. Now looking at group 1 and 3, right, we can see the only difference here is the tip in group 1 can detect light and the tip in group 3 cannot because it's an opaque cap. And group 3 does not experience phototrophism and group 1 does. So we can conclude from that it is some kind of light sensory response, right? So if we combine this, we know it's in the tip, we know it's a light sensory response. So we can give support for the claim that there's a light sensory response located in the tip, right? That cells located in the tip detect the light. Right, so we showed that it's the tip and we showed that it's the light. Okay, in treatment groups 4 and 5, the tips of the plant are removed and placed back onto the shoot on either a permeable or impermeable barrier. So the permeable barrier, things can get through, and the impermeable, things cannot get through. Using the results from treatment groups 4 and 5, describe 2 additional characteristics of the phototrophism response, right? And so let's look at what happens when you have a permeable barrier, right? And what happens is it's the same as the control, right? So having the tip there but disconnected and through a permeable barrier are the same, right? So what kind of pathway might this show? It appears as though there might be some kind of cell signaling pathway, right?
Yeah.
Where some cell in the tip detects light and then it transfers that information down the stem, right? So that's one characteristic that we can show. And we can show that that signal can get through this permeable barrier, right? Which is why there will be no difference.
Mm-hmm.
Now, we have an impermeable barrier, right? So the first thing we can show is it's through cell signaling, right? The other thing is, right, if we don't let things get from the tip into the stem, what happens?
The signal won't
Right.
be transferred.
So the signal is in the tip. We know that. We showed that in part A, but where is the response located?
Closer to to the bottom of it.
Right. It's in the bottom of this. So those are two characteristics that we can show given this response. And I'm actually going to just double check that those are the two that the AP would like us to acknowledge. So, yeah.
Sorry, I'm trying to look through this tiny phone.
Yeah. So it says, so the first thing it says is the tip produces a signal, right? Or some kind of a signal or a substance or a hormone, right? So that's the one thing. And then the substance must diffuse from the tip, right? It must get away from the tip and into the rest of the cell. So those are the two properties that the AP wanted us to acknowledge. Okay, let's look at 2013, sorry. And this is actually an example of an uncontrolled experiment. It's an experiment without a control, and we'll show how to work with that. And then maybe we can even identify what could a possible control be and what could a possible thing to look at be. But not every experiment needs a control. We'll see that. Right, because we can control, or we can compare, we can compare one frequency to the next. So I'll sort of explain this before we go down. These will be graphs of the absorbance of light by a plant in photosynthesis or some other kind of photosynthetic bacteria. We'll see what we're looking at here. And so we're looking at the wavelength of light that shines and the relative absorbance. And we have apparently two different kinds of photosynthetic organisms here. So let's look at what this says. An absorption spectrum indicates the relative amount of light absorbed across a range of wavelengths. The graphs above represent the absorption spectra of individual pigments isolated from two different organisms. One of the pigments is chlorophyll A, commonly found in green plants. The other pigment is Bacteriodopsin, whatever that is, commonly found in purple photosynthetic bacteria. Right? The table shows the approximate ranges of wavelengths of different colors in the visible light spectrum. So identify the pigment used to generate the absorption spectrum in each of the graphs above and explain and justify your answer. Right? So the thing we have to know about photosynthesis here is that a plant will absorb light, right? And whatever it doesn't absorb, what does it do? It would, if it doesn't absorb the light, it should probably reflect that light, correct?
Yeah.
So if we have a green plant, it will absorb very little green light because it will be reflecting the green light, right? Or if we have a photosynthetic bacteria that is purple, it will be reflecting most purple light and not absorbing purple light. So let's look at this. What we're interested in here is actually the wavelengths that are least absorbed, right? We're looking at what's reflected, so we want to look at what's least absorbed. So let's look at graph number 1. If we see there's sort of a minimum here, right? Here it gets away from the visible light spectrum, but there might be some kind of red around here. But let's look over here, right? Here's another minimum. Looks around 450 nanometers. And 450 nanometers looks right between violet and blue. And then we also have some red in here. And hopefully you know from probably elementary school, if we have violet, blue, a little bit of red, what kind of color would that make?
Violet, blue, and red, purple.
Purple, right? Even you could just sort of say that over here there's a minimum at violet, right? And violet is a shade of purple. But you could add in the red. I'm sure they would not take off credit for that, saying that it also reflects red. It reflects, so it's going to reflect violet, blue, and red mostly, right? Which will be purple, right? And so which bacteria would that be? Or which plant would that be? Would that be chlorophyll A or the photosynthetic bacteria pigment for graph
Chlorophyll
one?
A is usually green.
Right. Right, that's what it says here. So here we're reflecting purple in graph one. So this would be the Bacteriodosphine, right? Or however that's pronounced. Graph number two, right? It looks here, well, we definitely have a minimum right around 500, 550, right? Where is that in here? Well, 500 to 550, this is definitely in the green range, right? If we look at this. So this, because it has a minimum absorbance, right? At green, it's going to be reflecting green. So that would make graph two represent chlorophyll A. Let's look at problem B. In experiment, identical organisms containing the pigment from graph two is a predominant light source. Light capturing pigment are separated into three groups. The organisms in each group are illuminated with light of a single wavelength. 650 nanometers for the first group, 550 for the second, and 430 for the third group. The three light sources are of equal intensity, and all organisms are illuminated for equal lengths of time, right? Which is important because that shows that we're only changing one thing. The only thing we're changing is the wavelength of light. Predict the relative rates of photosynthesis in each of the three groups, justify the predictions. So the first thing we need to acknowledge is that the more light you absorb, the more photosynthesis is going to go on, right? Mm-hmm. And so now let's look at 650, 550, and 430. So let's start with 430. 430 here looks like it's right around a very high part, right? So this looks like 430 will actually have the most absorbance compared to 650. And then 550 should have the least absorbance, right? So using the absorbance and saying that the more light is absorbed, the more photosynthesis that goes on, right? We can say that at 430 nanometers, it will have the highest rate of photosynthesis, then 650, and then 550, right? Mm-hmm. And so that's what they want. Or they also said that they would just accept being told that so they said they would actually accept just being told that it says here that 430 and 650 have a much higher absorbance and a much higher rate of photosynthesis than 550. They say that they would give 4 out of 5 for that. So they wanted the rest of the ranking as well, but okay. So now let's go on to our last question that I have here, which is 2013 question 7. And it says here in an experiment, rats averaging 300 grams of body mass were tested several times over a 3-month period. For each individual urine was collected over a 3-hour period after ingestion of 10 milliliters of liquid. Either water, 1% ethyl alcohol solution, or 5% ethyl alcohol solution. The volume of urine was then measured, and the results were averaged for all the individuals within each experiment group. The data is shown in the table. So, propose one scientific question that the researchers were most likely investigating with the experiment. Again, the scientific question is going to be, how does the independent variable affect the dependent variable, right? Mm-hmm. So the independent variable here is what fluid is ingested, right? And so that might be the amount of ethyl alcohol in what they and what is ingested, and the dependent variable, the output, is the urine output. So here are the 2 questions that the College Board recommends, but they say they would also accept other sort of similar questions to this. So it says, how does alcohol consumption affect urine output in rats or any mammal, right? I could use rats sort of being an example of just a type of mammal. Or how does alcohol consumption affect the regulation of the kidney, right? Which is the organ that is going to be responsible mainly for urine output. Right, so that's the scientific question. State a hypothesis that could be addressed, right? It doesn't even have to be correct. It's just a hypothesis of a potential answer to that question. So maybe a correct hypothesis is if a rat ingests more ethyl alcohol, it will produce more urine. Right? Or you could have, so you could have a bunch of different hypotheses. So you could even say alcohol consumption has no effect on urine output in rats, which is incorrect, but that's a hypothesis. Or alcohol consumption reduces urine output in rats. Again, incorrect, but it's a hypothesis. It doesn't have to be correct. But basically we're taking a guess, a possible idea of what could be the answer to our question in A. Does that make sense?
Yeah.
Okay. Now C, using the data table, describe the effect of ethyl alcohol on urine production. So now we want the correct answer. And it's pretty easy to see here that the more ethyl alcohol you have, the more urine is produced, right?
Yeah.
So that's basically what they're looking for. So those are all the questions I have. I want to go back to this one though. Because even here, we don't have like an exact control, but we are definitely comparing the results, right? And same thing here, right? We don't have a control in this experiment, but we're just comparing, right? We're comparing different nanometers of light, and that is okay. But I want to give a challenge as to what if we wanted to make this a controlled experiment? What kind of question could we ask, and then what control could we add to make this a controlled experiment? And that's a fairly difficult question, but if you can get that, you will definitely understand the idea of a controlled experiment very, very well. I don't know if you have any guesses, Sam, or I can just go for what my answer would be.
I'd rather you give an answer.
Yeah. So what I might think is, what if you wanted to see, can you increase or decrease the rate of photosynthesis, right, by shining a specific wavelength of light at a plant or a bacteria, right? And so our control group that we don't have here would be white light, right, normal sunlight, right? And then we could see, well, how does a specific wavelength of light being shown at the plant, does that increase or decrease the rate of photosynthesis of that plant, right? Again, controlling, making sure we have the same intensity. So whatever light you use has the same intensity as sunlight, which I know there definitely are like lamps that can mimic the intensity of sunlight. So that would be a potential question. Maybe you have another one. If you have one, you can comment that below. But that's just one way that we could actually turn this into a controlled experiment. But not every experiment needs to be controlled. But I'd say, if we look at all of these, most of the experimental questions I've seen on AP Bio have been controlled experiments, but not necessarily, especially when we have like graphs like this, they're not necessarily always controlled. Okay, so hopefully that makes sense. I don't know, Sam, do you have any questions about any of these problems? Anything that's a little bit confusing that I could touch more on at the rest of this video, or I can end this video?
No, I think I'm good.
Okay,
Yeah,
well, thank you for coming, Sam. Thank you guys for watching, and I'll see you in the next video.